Good evening, everybody, and welcome to the second part of the pig microbiome for the Iowa Pork Industry Center monthly webinar series. We're pleased to have three speakers on the program tonight. The first speaker up will be Dr. Nick Gabler. Dr. Gabler is the inaugural John F. Patience Professor in Swine Nutrition in the Department of Animal Science at Iowa State University in Ames. He obtained his Bachelor's of Agriculture Science and PhD from La Trobe University in Melbourne, Australia. He completed postdoctoral research work at Purdue and at Iowa State Universities before joining the faculty at the Animal Science Department at Iowa State in 2008 as an assistant professor. We also have Dr. Laura Greiner. Dr. Greiner is an assistant professor here at Iowa State in the Department of Animal Science. Laura teaches undergraduate swine science courses and has an established research group that conducts research centered around addressing contemporary challenges in swine production. Also with us, speaking to us tonight about pig microbiome will be Dalton Humphrey. Dalton completed his master's degree in 2022 from Iowa State under Dr. Greiner. His work evaluated the impact of organic acids on the growth and odor emissions of grow-finished swine. Dalton is currently working towards his PhD and plans to graduate in May of 2024. So with that, Dr. Gabler, I believe you're up first, and please take it away.
All right, thank you, Chris. Sure. Yes, hello everyone, and welcome to this webinar that we're going to talk about with the swine microbiome. There it goes. Oh, so really what we're going to do today is just kind of do really a shallow dive into what we kind of know about the microbiome or microbiota in swine. Also, we'll highlight some opportunities and where some of the research is going in that, and then also I'm going to close up this presentation by giving some of my thoughts on what some of the next steps or where the opportunities are with regard to the pig and the microbiome. So really, if we're talking about microbiota, really that describes the living organisms found in a defined environment. And in this case, with regard to the pig, we could talk about what's found in the feed, what's found in the barns or on the floor. We could also more specifically, really what we're talking about is what's found within the gastrointestinal tract and the reproductive tract, or actually in the respiratory tract. The other thing about the microbiome can also be referred to as a collection of all microbes, including bacteria, fungi, viruses, and their genes. And so a lot of the talk today is going to be focused more on bacteria, but we can't forget that the contributions of the fungi and also the viruses, what they have actually on the microbiome as a whole. And so if we're talking about microbiota, and particularly for bacteria, a lot of the data that's out there, a lot of times people talk about bacteria at the phylum level. Then that can be broken down into the class, the order, the family, the genus. And then when we're talking more about clinical diagnostics or disease diagnostics, then we get more into the species and strains. So depending on the level of how they are identifying the microbiome or the microbiota is going to have has an impact on how we should interpret the data. And a lot of the data that people talk about and what will be presented here will be anywhere from the phylum to the genus level. So this area, one of the reasons we're asked by Iowa producers to actually talk about the microbiome is because it's a very complex area. People want to know, what should we know about it? What should we believe, or what's good, what's bad, what's indifferent? And the reason why this is a complex area, because we're really talking about the host's ability, i.e. the pig's ability to actually support other organisms in the digestive tract, or the gastrointestinal tract, the respiratory tract, or the reproductive tract. And then by harboring these microbiota, therefore there's a synergy between the host and the microbes. And at the end of the day, we want to make sure that the host actually can capture value actually from those microbes, whether it's directly from host microbial interactions or actually from the metabolites they produce. But this microbiota, or this population of microbes, are involved in many things to do with the host. They're involved in digestion, they're involved in host immune interactions or host immunity and tolerance. They're particularly important with health and disease, particularly on the onset of disease, whether it's clinical or subclinical, and then also in disease prevention. And the other area that's gained a lot of interest in the human literature is, or in the biomedical literature, is this whole gut-brain axis and the ability of the microbiota to actually work in a neuroendocrine manner and actually impact the brain, and how that it impacts actually activity and mentality and other behaviors. And so the gut-brain axis is a big opportunity where a lot of the biomedical research is going. And that's more to do with tryptophan and other amino acids being metabolized by the bacteria, and then for catecholamines and norepinephrine, dopamine, etc., all being produced by, all having an impact actually on the host, i.e. the pig. They can then impact a lot of different behaviors. Next slide then. Yep. Cool. And so with that, I'll hand it over to Dr. Greiner to talk a little bit more about microbiota.
Thank you, Nick. So I'm going to start in with just a little bit of information here on really big level, what do we know, and then kind of bring it down into a couple of different classifications. And you're going to see tonight that some of the data that we're covering is conflicting at times, and other times it's fairly vague. And as Nick alluded to, a lot of times we're basically finding papers where we're identifying the organisms, but maybe we're not lining them up fully with what that functionality is, or necessarily what the phenotypic response is, meaning what am I seeing in the barns. So when we first start looking at swine microbiota, the first thing we see is the phyla. And the phyla, of course, is kind of that general classification at the top, or towards the top of our list of classification pyramid. And what we notice is that the firmicutes and the bacteroides are the two that are the most predominant ones when we take those fecal samples from pigs. And the firmicutes appear to be the predominant phyla regardless of age in the pig. Interestingly enough, the population does appear to be higher when pigs are housed outdoors rather than indoors. Again, we don't always fully understand the reasoning why, but that is, of course, what's being found. And the bacteroides are actually the second predominant phyla. And interestingly enough, these are known as the obese organisms. And so if we look in mice and human literature, where we find higher populations of these organisms, those animals that have those higher levels tend to be obese. And actually in pigs, they do believe that's actually beneficial because it does correlate to higher growth rates. But again, these are things that we're again trying to explore and better understand what these different classifications mean. And so here's just an example. If we look across different parts of the digestive tract in particular, we look across the mucosa of the stomach versus the duodenum or the cecal mucosa and the feces itself, we see that those organisms are fairly consistent. There is a little bit of jumping around with the firmicutes in a couple of locations. But the bacteroides do actually kind of disappear out of a couple of those locations when we look at the duodenum and the jejunum. But again, they do account for about 85% of all the organisms that we can find within the GI locations. And the Proteobacter is about the only other phylum that we really can locate within the GI tract. And here's just another quick example. If we think about that suckling pig, it's going through a series of changes while it's still on the mom, potential environmental challenges, immune responses, as well as just antibody levels changing over time. And then, of course, the impact of weaning and that stress. And we do see that the firmicutes, while they do shift a little bit, do remain relatively constant throughout this process. So again, the question is if this doesn't really change, how do we impact it, or what can we do to alter the pig's performance? And so we're going to dive a little bit deeper into some of these categories, if you will. And the first place we're going to start is with the suckling and the weaned pig. And again, some of this is just a lit review of what organisms are we even talking about. And so we'll start there, and then we'll kind of dig into maybe some of the responses associated with these different groups or categories, if you will. So with the suckling pig or the weaned pig, actually in the genus category, we see the bacteroids were the greatest during the suckling period, and then the Prevotella increased after weaning. And so we do see in the genus category, we do see some shifting between a suckling pig and a weaned pig. If we go a little bit further down into the family category, we start to see things that might be a little bit more familiar to you, like Lactobacillus. And that is predominant after weaning. And one of the more predominant species there is Lactobacillus imliveris. And again, those are ones that we would expect to see in this time frame. Those are the ones we continue to talk about when we talk about nursery diets and trying to encourage Lactobacillus production. Those types of organisms would be in this category. After weaning, though, what's interesting is we do tend to see this decline in Lactobacillus organisms over time, as well as Enterobacteriaceae. But we do start to pick up Streptococcus suis in feces. And so again, when we start thinking about pathogenicity and maturity of the gut, all of these things are going to interplay with each other. And these are just, of course, data that demonstrate that this is changing over time. This is not specific to a diet, to antibiotics, or anything specific, but rather just general changes that we see within the gut. And so when we think even about that grow finish pig, as I mentioned, we see a lot of Streptococcus suis. And so the Streptococciae family is something that we're going to see a lot of. Lactobacillus is going to be second, and then we're going to move on down to the rheumatococcus and the Clostridia type of organisms. So again, you sit here and you can look at this and go, well, what do I do with this information, and how do we interpret it, and what does it really mean? And so what we do know is that, again, as Nick alluded to, some of these organisms are going to be specific within different parts of the tract. Lactobacillus is actually abundant throughout the tract, as well as Clostridia. And so when we think about feeding these organisms, we can recognize that there is potential to change performance across multiple parts of the tract based on what we're seeing. The microbiome itself, though, the microbiota, they're going to be influenced by a variety of things. I've alluded to a few of those. Certainly, anytime we stress that pig, whether it's through weaning, through a transport, through mixing into a new population, that can cause a shift in the microbiota. Obviously, mom can transport various microbiome organisms to her offspring, whether that's through nose to nose into the respiratory microbiota, through the feces into the gut, and even through the colostrum. Feed, we know, has a huge impact, and we'll spend quite a bit of time on feed tonight. Temperature, so for example, it's really hot today. That alone can be changing the microbiome itself, as well as things related to health, such as disease and antibiotic usage. And so all of these have this interplay. And while we know that we still relatively preserve the predominant groups within the gut, the question is, well, what are the small changes? And do those small changes, in fact, change performance? And so let's talk a little bit about that. The optimal microbiome in this situation for growth performance is likely dependent on a multitude of environmental and host factors. So we cannot sit here today with our presentation and say, you need to have this population of Bacteroides and this population of Streptococciae and this of Lactobacilli. That does not correlate to anything in terms of growth performance. And so it has to be kind of this marriage, if you will, between what the pig's experiencing through its environment and what's within its gut to optimize its performance. What we do know is that if we look at carcass and growth merit, that there are a few groups that do appear to have the most influence, and those would be associated with the firmicutes, the Bacteroidetes, the Proteobacteria, and the Actinobacteria. And so again, those are some organisms that we can start to focus on when we think about doing research studies or looking at our influence of nutrition and how we influence those populations, because those do tend to look like they have more impact on growth and performance compared to some other classifications or groups. What we do know if we start breaking those apart is that the firmicute group is involved primarily in maintaining energy balance within the animal. And so we can think about that associated with short chain volatile fatty acid production and fermentation processes. The bacteroid group is more involved with butyrate production, again with fermentation, and then in turn also will activate T-cell mediated immune responses. And we talked about that in the last webinar series session where we went through and we showed you a picture of all the different things that short chain volatile fatty acids can do to the animal. And it's not just at the gut level. That can actually influence things such as, lung inflammation, glucose regulation, and so forth. And so this population has a very significant impact on not just what's happening at the gut level, but well beyond the gut level. The Proteobacter group is more involved with GI health and can actually be involved in triggering inflammatory responses. And so what I found was actually interesting when you look in some of the data. If we dig a little deeper, we can actually find that non-pathogenic E. coli levels and Campylobacter levels, if they're relatively high, can result in better survivability during PRRS infections and can increase weight gain. And again, we talk so vaguely a lot of times about E. coli and thinking E. coli is bad, but not all E. coli species are bad. And in fact, in this case, there's nice data that would suggest that having certain types of non-pathogenic E. coli could actually be beneficial to the animal. The same with things like Prevotella, that can actually help improve intermuscular fat and storage of glycogen in the animal. Lastly, if we think about the Lactobacillus species, which are the ones that we talk a lot about in the nursery, those are more involved in increasing natural killer cell activity and increasing mucosal antibody production. So we get that better gut health and protection there at that barrier. So now we're going to kind of step into just a few things we know about different components of nutrition on the microbiome and the microbiota itself. We are not going to focus on specific feed additives. We're not going to talk significantly about the feeding of probiotics in this situation. We know probiotics have potential for influence. We're going to focus more on things that we might do in other parts of nutrition that could have an influence on performance. So one of the things that we do see is that when we feed things like fish meal, soybean meal, or soybean meal with cottonseed meal, we can see this greater bacterial diversity in the data sets relative to just feeding like cottonseed meal. In those situations, we see firmicutes as the predominant phyla, and then Proteobacter actually became a more prominent phyla when we fed fish meal. And then we saw that it was actually E. coli, Escherichia, and Shigella species that increased. Lactobacillus, though, became more abundant when we fed cottonseed meal. And so, again, different proteins are going to influence different types of responses. This shouldn't be a surprise. We talk about feeding soybean meal versus other types of proteins in the nursery pig, particularly right now as we're dealing with different types of E. coli and what that could do for an impact. But what I do think is important to note here is that we find a lot of studies that identify organisms, but these organisms don't always correlate to disease or protection or performance. And in fact, we might actually see that one study will have one result and another will have the complete opposite. And so interpreting data can be very, very conflicting. In addition, when we think about protein diets, I just mentioned soybean meal, but if we feed high levels of crude protein, we can increase the coliform counts in the feces, which actually can increase PWED incidence. Whereas if we feed lower crude protein levels, we actually reduce the diversity of the colon digestive bacteria after an E. coli challenge, and that increases butyrate producing bacteria. So in one study, I just told you that feeding cottonseed meal was a good thing. It increased diversity and it produced more beneficial organisms. But in this study, we're seeing that lower crude protein decreased diversity after an E. coli challenge, and that actually allowed for improved performance. So this is where that conflicting data can come into play, is we don't always understand what's happening with those short chain fatty acid production events. If we switch over to fat, I think fat's another interesting one. It may not be one that we think much about when we think about the microbiome. Most of us tend to think about protein and fiber in particular, but fat does have potential to influence the microbiome. High fat levels in the diet can actually decrease the diversity, and we can also see this increase in the firmicutes, but a decrease in the Bacteroides. So firmicutes, as we've talked about in the past, could have some benefit, but so could the Bacteroides. So the question is which one is going to be more influential in terms of growth performance? And in this study, that data was not provided. And so we don't really know what that outcome means. We just know that feeding high levels of fat can in fact change that diversity and change the populations. When we look at omega-3 and omega-6, we can see that omega-6 increases inflammation-prone organisms. So omega-6s can increase Clostridia and Enterobacter in the gut, where omega-3s will suppress inflammatory organisms while promoting Lactobacillus and Bifidobacter. So again, we have to think about omega-6s versus omega-3s and the potential benefit, not just at the pig level, but then also what could be influencing within the microbiome, and then how would that also be influencing the piglet's performance. As I mentioned, most of us like to think about fiber when it comes to the microbiome, and I think that's a very fair discussion. Certainly when we think about converting non-digestible fiber into energy, we do rely on that microbial population to do so. We do know that organisms like rheumatococcus can be found on wheat bran and resistant starch. And then while they're there, the Eubacterium rectal will convert that acetate to butyrate. And then of course, that's something the pig can use for energy for those inflammation responses and immune responses that butyrate's involved in. Feeding high fiber diets certainly does increase the lactobacillus abundance in the small intestine and those volatile fatty acids in the hindgut. Whereas feeding insoluble fiber like DDGs can reduce the firmicute to Bacteroides ratio and lactobacillus. And I would say years ago now, I guess I'm going to date myself, when there was a concern of seeing some brachiospiro dysentery through the US. Certainly there was concern around feeding DDGs and the potential to allow brachiospiro to proliferate. And so we certainly do want to be thinking about the impact of insoluble fiber and how that could actually create populations that are not beneficial to the pig. The other types of fibers that we might be thinking about would be, pea, maize, and wheat bran, and those will, of course, improve the lactobacillus and the bifidobacter. When we're dealing with E. coli issues, maybe feeding a wheat bran or a soybean fiber, a wheat bran would reduce the E. coli, a soybean fiber would increase the E. coli. And so again, it's important to know what type of issue you're dealing with within your barn to decide what type of fiber is going to be beneficial to the pigs. Here is a really interesting little paper that I found recently looking at two different fiber sources. So looking at sugar beet pulp and soybean hulls, and then comparing that along with soy oil and choice white grease. And so there were no differences in average daily gain or feed intake. The only thing they found in terms of the microbiota was that there was an increase in this Reproductive in the colon. But what was interesting then with that is they did see a fiber response, and that was all they saw. They did not see a response to feeding a different type of fat, and there was no interaction. And so this group was trying to relate the response to feed efficiency and fiber to this increased level of Reproductive in the colon. There probably needs more work on that to determine if that is in fact correct. But nonetheless, it was an interesting study to start to talk about the influences of multiple factors that we're doing within the feed, not just fiber, but now I have fiber and oil, and don't forget we'll have protein in there. And so how do all of those interplay within the microbiota, and what does that mean for the phenotypic outcome of the pig? I am going to talk a little bit about enzymes in here. I did think it was important to talk about enzymes, particularly when we think about enzymes that can help break down non-digestible fiber and other potential nutrients in the body that the pig can't effectively use very well. In this group, Amy Petrie had done some work looking at high fiber diets and adding xylanase. When she added xylanase, you can see there was a significant increase in a handful of different organisms. Bifidobacter, which we've talked about, actinobacillus we've covered, as well as lactobacillus. And so some of what we would consider more beneficial organisms, and it reduced the Streptococcus and the Tersibacter in the ileum. In addition to that, it decreased the Clostridium and the Escherichia. And so in this case, we would say that feeding xylanase made the gut more favorable, although we don't know if those Escherichia organisms were the non-pathogenic or the pathogenic. And so again, remember, if it's non-pathogenic, we could actually be improving performance. And so this is where the data that we have at hand isn't always very clear as to what we could be actually seeing phenotypically. Phytate is certainly another one that many of us are concerned about. We know it's an anti-nutritional molecule, and many of us use phytase to liberate that phosphorus from the phytate molecule. And if we look at phytase in particular and its relationship to the microbiome, we do see that supplementing phytase into diets can increase Bifidobacter and Clostridium numbers in the ileum. And so again, here you go, well, there's one what we think is a beneficial population and one that may not be beneficial. So we don't really know what those implications are on gut health or performance. And so again, this is just, sounds a little confusing, but this is really what we want to demonstrate in some ways, is that this is not a clear cut, we feed this and we get this response to the microbiome, and then this is what it means to pig performance. So I'm going to switch gears and I'm going to step out of nutrition and I'm going to talk about gut health and the microbiome. And again, just to remind you that, we're focusing a lot on butyrate, and it comes from fermentable carbohydrates that increase short chain fatty acid production. And of course, the one we're talking about is butyrate, and that's involved in mucin production, reduces bacterial adhesions, improves immune function. And we've been talking about lactobacillus, and lactobacillus, of course, is involved in increasing mucin production and creating that barrier. And so when we look at weaned piglet diarrhea in particular, one of the things that we notice is when pigs are going through a round of diarrhea, there are reduced levels of lactobacillus of various species, as well as an increase in the pathogenic E. coli. Some of the data that's out there related to feeding nursery pigs, and we've alluded to this already in the discussion around fiber, is that using things like wheat starch and beet pulp can improve lactobacillus. And this is pretty documentable. This is pretty repeatable data here. We also know that feeding fermentable carbohydrates, resistant starch, hemocellulose, those can also all increase lactobacillus. And so while I maybe just confused you with a whole bunch of slides, there are some things that we know we can nutritionally do to hopefully push some of these populations up, to hopefully give that piglet a better chance of producing more mucin and fighting against that E. coli infection. Another paper that we've talked about in the past has been around Nair et al. And this group was actually looking at mycoplasma exposures to pigs, and they were looking at the microbiome at the same time. And what they discovered was that in the pigs that did not appear to have significant lesions after the mycoplasma exposure had elevated levels of rheumatococcus in their feces. And rheumatococcus, again, is one of those organisms that can be involved in producing significant amounts of butyrate. And so we do see, again, that this butyrate then moves on, and it can, as we know, improve lung functionality, reduce inflammation, and really get the immune system moving in the right direction. And so this paper does document that if we do have issues in the lung, there's potential to change the microbiome in the gut that could then have a positive outcome systemically on the animal. So one way to improve or increase rheumatococcus within the gut is to feed things like resistant starch. And so again, if you're thinking about dealing with a PRRS exposure or an influenza challenge or even a mycoplasma in the barn, it is possible that you may want to consider finding ways to improve the amount of resistant starch that you're feeding so that you can improve those organisms and potentially improve the performance of those pigs. We say potentially because, again, the data is, there's not a lot of data out there to support that in fact is going to maybe fix it all, if you will. Enterococcus facilis and other filamentous bacteria can actually be involved in increasing the response against pathogens such as porcine reproductive respiratory syndrome virus. So not necessarily rheumatococcus in this study, but enterococcus. And so again, if we can find ways to promote enterococcus development and growth within the gut, the pigs may have a better chance in fighting against PRRS. We also note that there are some studies that would suggest that lower microbial diversity would result in poorer performance when pigs are infected with PRRS. And so again, this might be some things that we need to explore as we go down the road. Maybe in some situations, a lower microbial diversity with the right organisms being the lead would have better benefits for certain health challenges, where others might require a greater diversity with different populations of organisms present. Here is a little study where they did a microbial transplant. So they took feces from a boar stud, if you will, and they exposed that. They gave that directly to some pigs, and then they exposed them to a challenge. And what they noticed was that when they did that, they actually reduced the lung lesions that were present in those animals. And so again, this is another example of different ways to use the microbiome to potentially manage disease. And this was referring to fecal transplants, which is something we're not going to spend a lot of time talking about in this presentation, but it is something that has been documented in a couple of studies now that can have some benefit on potentially compromised populations. So now I'm going to turn the slide set over to Dalton and let him talk about sows.
Thanks, Dr. Greiner. So I'm going to take a few minutes to talk about sows and their relationship and their microbiome and how it may impact multiple pieces of production. And so we really see as the sow is a focal point of somewhere that we can really impact multiple phases of production due to its implications, not only on maternal health and reproductive performance of the sow, but also neonatal health and opportunities to set that piglet up at birth for success long-term. If we go back to that large-scale study Dr. Greiner mentioned in the beginning, looking at associations with OTUs and performance, those same authors showed that those OTUs associated with performance actually had varying levels of heritability. And so there was an association between the sow side of things and those OTUs specifically associated with performance. And some of those ranges were very low, some 2.5% heritable versus all the way up to explaining 55% of some of the variation in those levels of organisms in the piglets. But the sow is also one of the first times in terms of age of animal where other locations become particularly important when we think of the microbiome. And so when we think of the vaginal tract being an important location, Wang and others were some of the first to characterize this in sows, and they saw similar to the gut, where it's primarily dominated by firmicutes, proteobacteria, and bacteroides, but they also saw that elevations in some of those or changes in those distributions of those organisms could be associated with health conditions like endometriosis. And so that brings up the question is, how do you interpret data when we see overlaps between healthy individuals and then diseased individuals? And so that really brings up 2 points to that. Here we're talking about, again, the phylum level, the really high level classification of those organisms. And that brings us to needing to look at more lower level genus and species and strain level to really tease apart what's happening and what may be more specifically associated with that, but also the concept of this broader community of organisms that's really driving some of these disease and normal states. And it may be more of a dysbiosis event that's causing this rather specific bacteria. So when we do look more closely into the genus level of some of these, they saw that there was associations not only with the vaginal microbiome, but also the intestinal microbiome. And so seems to be connection between the gut and then larger reproductive performance in the sow. So outside of disease conditions like endometriosis endometriosis, there's also been authors associating different microbes with other reproductive parameters like estrus. And so Wang and others in 2020 saw that there were certain bacteria associated with delayed puberty expression in gilts, rheumatococcus, lactobacillus, but also other bacteria that were associated with more normal puberty expression, Prevotella, Treponema. And so we do see that there are different organisms associated with very specific parameters that are important to us in production. But if we take a little bit more, a step more down the line and think of neonatal health in the sow, we've for a long time known about this connection between the maternal gut and the mammary gland, this enteromammary axis. And for a long time, it was mostly associated with things like immune cell transfer and early passive immunity to the piglet, but it's also becoming evident that this may be a pathway for bacterial populations to establish themselves in the neonatal gut. This concept of an enteromammary pathway, where bacteria may be picked up by immune cells in the maternal gut, transferred into the blood, and then ultimately deposited into the mammary gland to be excreted in milk. And then those bacteria may be an early population that could be establishing themselves in the piglet gut, along with environment, fecal transfer from the sow, and those sorts of things. And so there may be an opportunity to manipulate that pathway to help set the piglet up early in life to have a more beneficial microbiome to support health and productivity long term. But we also know there are differences in terms of age of females and their microbiome in terms of gilts versus sows. And so that's something that we need to be cognizant of as we think of this connection between the maternal gut and then ultimately the neonatal gut. And so our group here has investigated this pathway in sows. And so we took samples from the sow, fecal samples, blood samples, and colostrum samples, and then sequenced those to look at consistency across samples and if there were shared organisms across those, indicating this route of transfer. And so this table just shows a quick example of some of the top 10 of those organisms that we did see shared across all sample locations in the same individuals. And so here we see that Clostridium sensu stricto 1 was present in all 3 sample locations in 15 different animals. But at least numerically here, we see that there is some obvious differences where only 2 gilts had that organism shared across samples, or 13 sows did, so indicating some differential transfer between parodies. And so overall, we saw that there was around 10% of these OTUs that were shared across blood, fecal, and colostrum samples, indicating there's a wide range of organisms that may travel through this pathway, and thus may be important things for us to be cognizant of as we either manipulate the sow microbiome or that specific pathway to help set up the neonatal gut. But when we think about those differences between sows and gilts that I alluded to, we did have a few organisms that we did see significantly different between sows and gilts that could also be important thinking about neonatal health. And so a couple of those being Clostridium sensu stricto 1, which I talked about in that initial table. Mycoplasma we saw elevated in all 3 sample locations in those gilts. And so we know this farm was mycoplasma stable. And so it's likely those gilts were serial converting at that time or had circulating mycoplasma. And so that wasn't necessarily a surprise to us, but does indicate that this pathway could be a route of transfer for not only beneficial, but also pathogenic organisms. Lactobacillus was higher in sow blood, but then we saw that in colostrum, it ended up actually higher in gilt colostrum. And then kind of thinking back to the trial Dr. Greiner mentioned, or the paper Dr. Greiner mentioned, rheumatococcus 2 was higher in gilts compared to sows. When we think of that connection that Dr. Greiner talked about between the lung lesion scores and rheumatococcus, And this elevation of mycoplasma and gilts in this trial, as well as rheumatococcus and gilts, so it could be this synergism going on where it may be that rheumatococcus could be supporting the neonates through this potential mycoplasma transfer. But largely when we think about this pathway, there is a lack of data largely on the impact of maternal factors like nutrition on the milk microbiome, how that may long-term affect neonatal immune development and performance, then also the mechanisms that regulate that pathway and which bacteria are able to take that route of transmission. And so we think that future research in terms of the sow microbiome related to this should focus on manipulating that pathway, like I alluded to improve both maternal and neonatal health. And we are currently assessing the impact of a fecal microbiome transfer on that pathway from sows to gilts to see if we can improve that route of transmission from gilts. So when we think again about some of the other issues we may see in terms of maternal health, Jason Ross's group here has done a lot of work with prolapses and trying to understand what may be associated with higher prevalence of prolapses in our sow populations. And they recently did a study where they took sows that had been perineal scored, which is a scoring system they have shown to be associated with risk for prolapse, and then characterize those microbiomes to see if there were differences or associations with a higher risk of prolapse. And so outside of characterizing the vaginal microbiome here, which you can see on the left side, they saw specific differences where sows that were at lower risk for prolapse had a lower abundance of a few of these key organisms like Streptococcus disgalactiae, treponema. And so there does seem to be some associations between specific organisms and major reproductive issues like prolapses. And so these two tables just show some examples of some of the more prevalent organisms, both on the genus and the phylum level, between these two different scores where the dark bars indicate a higher risk for prolapse and the lighter bars, a lower risk for prolapse. And then aside from specific organisms, they also showed here on the left side of this plot that there was a more broad separation between these groups. When you think of it on a whole community level, they saw clustering and differential expression or structure of the community. And so it again indicates this isn't necessarily a specific organism thing, but more a community-wide event that could cause dysbiosis and issues from a health standpoint. So with that, I'm going to hand it over to Dr. Gabler to talk about next steps.
So before we get into next steps, just want to highlight, I think, one of the major areas where I think the microbiome can actually be useful in the sense of pig production. And that's really with regard to decreasing or modulating the risk factors associated with disease. And this is, Dr. Greiner alluded to this, but this is some work that Dr. Emma Helm did in collaboration with Eric Burrow at our Iowa State Veterinary Diagnostic Lab. This is where we actually inoculated pigs all with swine dysentery or brachyspire dysentery and gave them swine dysentery. And over a 42-day study, we fed these pigs 3 different types of diets, where one was really a low fermentable carbohydrate diet, in other words, made up a lot of mainly corn distillers grains. And then we switched out the corn distillers with higher levels of actual resistant starch or resistant potato starch or sugar beet pulp. And so as we increased the concentration of fermentable carbohydrates in the diet, these pigs became actually somewhat resilient or robust to the actual swine dysentery challenge. Even though that the pigs that were fed the high fermentable carbohydrate diet, all 11 out of the 13 of them came back culture positive, fecal culture positive for brachyspire dysentery. The only 2 out of the 13 pigs actually showed clinical signs of disease. And then we did some sequencing of the fecal sequencing of these pigs, and we found that the higher amount of non-fermentable carbohydrates in the diet, they had a greater relative abundance of anaerobes. And in particular, what we found is that Provotella is actually was found as more of a predisposing bacterial species for swine dysentery. So a higher amount of Provotella actually in the pig, we seem to have a greater degree of clinical disease in these pigs. So I think disease prevention, or at least mitigating or taking the edge off clinical disease, is one way that we can modify the diet, that you can change the microbial populations, and therefore that can be a really good way to actually help modulate that. But really, what's some of the next steps, or where do I see the opportunities for that? Is really, to be honest, we don't know, no one really knows what the ideal microbiome is. What should we really be achieving? This is going to be dependent on what your production goals are, on the genetics of the animal, the location, what part of the world, what climate you're raising your animals in. Are they on slats? Are they in pasture? What's the disease pressure? What's the antimicrobial use? All of these are going to be factors that are going to alter the microbiome. And one thing we've got to remember is that the microbiome, there's a lot of redundancy actually in the microbiome. Different phylums and different species and genre can all increase or decrease. But then what we really need to know is, what are they actually producing? And so the microbiome, we know that we can modulate the microbiome through diet, through stress, through disease, through fecal transplants. Even more recently, there's been work that's shown that we can use vaccines. We can actually change the microbiome by vaccinating against Salmonella typhimurium. There's other antimicrobial agents that can be used. And so all these can be used to manipulate. But what are we actually trying to achieve? Next slide. Yeah. So really, what I think that one of the biggest areas that we need to focus on is, what is the microbial population producing, rather than what phylum or what genera are there. Most of the papers that are actually published on the pig microbiome fail to actually show any host-specific effect. In other words, what's going on with reproductive performance, lactation performance, growth performance, any kind of host-related production parameter. You don't really see a lot of that. Yet they always show that the microbiome can be changing at the phylum or genre level. And really, so what that's really telling us is that there's redundancy in that microbiome. And so until we know what is being produced and what type of bacteria, what kind of species and strains are actually producing certain types of metabolites, whether it's short-chain fatty acids, bioamines, etc., and then how the host actually utilizes these, we really aren't going to make a lot of inroads into what the ideal microbiome is going to be. And so we need to move away from phyla and focus more on species and strain specific or specificities. In other words, what's actually there and what are they producing? And so the greatest opportunities to enrich and modulate the microbiota I see is really in early disease prevention, or in early life of an animal, to actually enhance growth performance and reproductive performance. And one thing I think we also got to take into consideration, it normally takes anywhere from 7 to 21 days for that microbiota to change to something and then actually stabilize to its new colony, or to its new communities. And so really, if we're going to change the microbiota, it's going to be generally a longer term game. And so we have to have some patience with that. And so, Yep. And so really, I think the last thing I'm going to say about the microbiome, there's a lot of opportunities in swine production, but also we've got to be very careful of the technology. And so depending on what type of sequencing you're doing, you're going to get different results. We've had it in multiple cases that we have actually challenged pigs with a certain pathogen. We've done some deep sequencing on that microbiota, on the microbiome, some metagenomics, and we cannot find the pathogen even though we've cultured it out. So depending on the reference libraries that you actually use to actually determine what microbiota is there, you can get different results. And I think this is one of the biggest reasons why we see such variability across from study to study, because we're always using different reference libraries, or people are using different reference libraries in different institutions or different parts of the world. And so that's where there's a lot of it's very gray, or it's very we're not sure exactly what is the ideal microbiome. But there is a lot of opportunity, and the biggest opportunity, I believe, is actually in setting up that pig for actually more disease resilience, where they don't actually see a lot of kind of clinical health challenges. And with that, I know it was a very short kind of prelude through the microbiota of the pig, but I'd like to thank you for your time, and we'd be happy to take any questions.
Yeah, Nick and Laura and Dalton, thank you so much. Really appreciated the practical look into the microbiome and things that you can do and what we're learning from the literature, and certainly agree it's a very complicated topic, and as we get more of these new tools out there, it's the more we know, the more we understand we don't really know. So I appreciate all your guys' work in there. Just a reminder to any of you in the audience, if you want to go ahead, and if you have a question, feel free to drop it in the chat box here, and we'll go ahead and get it to the presenters. I do have one question for the panel here, and really interesting information based on some of the lit review stuff that's done, and knowing how we've kind of seen an increase in post-weaning E. coli. I was kind of wondering, and I saw some of the information that Laura was sharing about different levels of soybean meal and how that was contributing to different populations of bacteria in the microbiome. Do you think, is there anything that you've kind of learned in that lit review as you guys went through all those different papers on microbiomes that would maybe challenge the way you think about formulating early nursery diets, particularly in pigs that are struggling with some E. coli challenges?
Well, based off data that we've generated over the last couple of years, whether we change the source of fiber to be more fermentable or less fermentable, so whether an oat groat based carbohydrate versus a soy hull versus a wheat bran, we do not see any difference with regard to a F18 ETEC challenge situation in a commercial conditions, or with high or low crude protein diets with regard to exacerbating or mitigating a hemolytic E. coli challenge. But the issue is, a lot of times, at least here in the Midwest, a lot of the enteric disease pressure is also confounded by the fact that majority of these pigs are all positive for rotavirus A, B, and C, and they've also got some other stuff as well. So it's not just a clean, at least a bacterial challenge, or at least bacterial disease pressure on these pigs. But I don't see any changes really, whether we increase our in a controlled manner, if we change that carbohydrate source to increase certain types of fermentation. I don't see any benefit or any positive or negative effect in these pigs. And we've repeated this a couple of times now. We just don't see it. So I don't know what else.
No, I think that's very fair, right? Some of the data would support, changing your carbohydrate source, but again, it's not always consistent when you get into the field. And so, as you know, that's some of the challenges we face with controlled studies in a clean barn versus putting it out in the field, that there's other factors that could influence the response that we're seeing. And I would agree, the data doesn't necessarily support what we see in the barn.
Because what's interesting, we see that we can use at least fermentable carbohydrates in a collective term, slang term. We know that we can really do a good job with swine dysentery or brachyspire challenges. We have not seen the same effect with Lawsonia intracellularis or hemolytic E. coli. So I think there's some nuances with how these pathogens interact with the fermentable products, or these metabolites in the lumen and at the epithelium that I think we need to know more about. Why does one work and not the other? And then it's not as simple as just saying, oh, we increase lactobacillus, we increase VFA production, and then that's going to be good for enteric disease, because it's not always straightforward as a one-to-one benefit. So it's horses for courses, unfortunately.
Do you think some of that has to do with where the samples are collected? Are most of the enteric samples, are those mostly rectal swabs? I'm just trying to think of brachyspira being more of a large intestinal disease compared to E. coli being more of a small intestinal disease. Does that have anything to do with it, you think?
think that's part of a lot of the published data is taken out of fecal or rectal swabs, fecal samples or rectal swabs, and so then we also people try to then relate that to regional specificities, and then luminal versus epithelial, mucosal associated are different again. And so I think like the chart the figure that Laura showed, there is differences between mucosal associated, luminal associated, and fecal associated. There is nuances that change. And it's even more profound, those changes, when you start getting down below down to the genre, or even down to the species level, they're going to be different again. I think that's what we're missing and need to know more about.
Yeah, no, fantastic. Couldn't agree more with you.
Yeah, I think we just spend way too much time just doing shotgun, like let's just measure it and see what's changed, and then not being able to then relate that to anything. I think we just need to be much more specific in what we're after.
Yep, very good. I don't see any questions coming in, so I think with that, we'll go ahead and thank our presenters, Dalton, Laura, and Nick, for contributing the great applied knowledge on the microbiome so far. And I want to thank everybody for attending. The microbiome will have the Iowa Pork Industry Center. We're continuing on with the monthly webinar series. The next one, will be more information coming. Dr. Schultz will provide a market outlook for us. Be looking for more information coming out with that. You can always visit the Iowa Pork Industry Center website at ipic.iastate.edu. So with that, we thank you very much for your attendance and have a pleasant evening.